Our remaining eukaryotic supergroups fall into two big categories. One of them is called the SAR group, the alveolates, Stramenopila, and rhizarians. And the other group is called the Uniconta, or the amoebozoa and opisthecants. So these two groups are separate groups, but the evolutionary relationships within those groups are still a little bit fuzzy. So depending on where you look, they might categorize them as one like super duper group, or they might categorize them as smaller individual groups. I'm not super worried if you know how they are directly related to one another, mostly just that you understand that the SAR group are more closely related to one another than to the other groups. And the amoebozoa and opisthecants are sometimes thought to be more closely related than other groups. Sometimes they are put into their own separate groups. But in total, you do need to know 7 supergroups. These 5 plus the Archaeoplastida and the Excavata that were in the previous lecture video. Our alveolate group, or the alveolata, are named for sac-like vesicles along the outside of their cell called alveoli. You might recognize the term alveoli because those are the name for the sacs inside our lungs that fill up with air when we breathe in. If you have asthma, the alveoli in your lungs are what become inflamed, which is not really important here, except that they are named the same, and they are both sac-like structures that are generally filled with something. So the outside layer of these alveolata are made up of these alveoli around the edges. And then some of these guys, like this picture down here, this dinoflagellate, is also potentially photosynthetic with a plastid inside. We've got an image up here of an electron microscope image showing the nucleus, the plastids inside, and then a cell wall. So some of our protists, even though they're not related to plants, also have cell walls. So they've got all kinds of things going on in our protist groups. So there are 3 important phyla. We're only going to talk about 2 of them. There's the Ciliophora. These are ciliated alveolata. These are not the same thing as some of the other ciliates that we might see. Remember, we have lots of things going on in our group of protists and lots of things that come up multiple times. Then we've got the dinozoa, or the dinoflagellates. These are generally going to be photosynthetic, but some of them are not. Dinoflagellates are really cool because some of them will bioluminesce. So we've got some cellulose here that appears blue. This is not bioluminescence. That is a stain that we put on. But if you've ever seen videos of bioluminescence happening in water, it is because of dinoflagellates. So if you just Google bioluminescence, you'll see all kinds of examples of waves and boats swimming through water, people touching water that cause these dinoflagellates to luminesce, basically to glow, and it makes a really pretty blue color. In a previous video, we talked about the red tide blooms. Sometimes our organisms undergo eutrophication, which is where our water systems undergo eutrophication, which is where we add in a nutrient, usually due to some sort of agricultural runoff, and then it causes the organism to bloom. So if you want to see a picture of this, you'll want to go back to the informal description of protist habitat, and we talked about it when we talked about the plankton organisms and the phytoplankton organisms. But sometimes when that happens, it is due to dinoflagellates. Our next group in the alveolata supergroup is the apicomplexa. The apicomplexa group is important because it contains some important parasites. We saw parasites back in the Excavata group. Our examples there were Trichomonas and Giardia. But we have another one here, along with a few others in this group that are parasites to human cells. So Plasmodium falciparum is part of the Apicomplexa group of the alveolates, and this is the malaria-causing protist. So the malarian protist is spread by mosquitoes that move the infection from one infected individual to another through transfer of blood between the individuals. So the mosquito bites the first person, draws up some of their blood, and then when they bite a second person, can release some of the infected blood cells into the second person. The malaria disease infects the red blood cells, which travel to the liver and then causes problems in the liver. So it can cause basically inflammation of the liver, produces red blood cells, and then it continues through its life cycle. So you can see this little curly Plasmodium organism here. It has kind of that spiral shape that we saw in our bacteria. And you don't necessarily need to know all about the life cycle here, but you should know that this important organism is part of the Apicomplexa and that this group has other important parasites. We're not going to cover those parasites right now, but we're just kind of hitting the highlights for some of the better known organisms. Our next supergroup is the S part of the SAR category. This is the Stramenopila. This one has a really diverse type of organisms. We've got plant-like, animal-like, and fungus-like protists all in this group, but they are all characterized by a specific structure on their flagella. So they have flagella, and on those flagella are fancy little hairs that work kind of like cilia. They basically beat like oars to increase swimming efficiency. So it makes them better at swimming in their environment if they are, in fact, in an aquatic environment. Now you might be thinking, well, this doesn't look anything like this algae over here. Remember that this is a common characteristic that is going to come from a common ancestor. So it is very, very likely that the common ancestor of all these different types had this flagella with these hairs on it, even if all of these aren't reflected in having that flagella as well. So some of these are heterotrophic, like the Phytophthora infestans, which causes potato blight. So if you are familiar with the Irish potato famine in the late 1800s, a lot of people died in Ireland, spurred immigration over to the United States because of a lack of food. This was caused by this Phytophthora infection. We've got a couple other ones. We've got some diatoms here. Diatoms have a silica shell, so this is basically a little glass shell. They come in all kinds of crazy shapes and sizes and colors. I don't know if this is colorized in a particular way or if it's lighting, but they look really cool. We looked at diatoms in our lab as well as an example of the Stramenopila group or the SAR group. Then we've got this brown algae or kelp. This is not a true algae like we see from the Archaea plastida. This is a separate group that has similar characteristics. They are photosynthetic as well, but they look very different than our green and our red algae, and that is because they come from a different evolutionary group. The R in the SAR category comes from the Rhizaria group. There are two major groups, the Radiolaria and the Foraminifera. We looked at Foraminifera in our labs. This image of the Foraminifera here doesn't really look like the one that we saw, and that's okay. Remember that these are really big phyla, and so there are a lot of species that can fall under a single phyla. But the common characteristics of these two groups are that they have mineralized shells. So these are not little insects. These are not clamshells or silver dollars. These are rhizaria that have a shell similar to some of our clams, sand dollars, things like that, which are animals. These guys have this mineralized shell, and then they also have something called Phylos pseudopodia. So remember that pseudopodia is a structure that extends out. It is a cytoplasmic movement of the basically one region of a cell that is going to kind of move out, and then it basically allows the organism to sort of creep along. So the rhizaria have a special kind of pseudopoda called Phylos pseudopoda. Basically, you can think of it as like a filament and long strand that kind of look like these spikes coming off. So they have these hair-like extensions that come off of them, which unites them into this similar group. Some of them are photosynthetic because they have a symbiotic relationship with algae. So this is photosynthesis based on a relationship, not necessarily secondary endocytosis, but a common function where they are coexisting together. All right, our second to last supergroup is the amoebozoa. If you look at your study guide handout, we'll see that the amoebozoa and the next group, the opisthokonts, are organized into a category called the uniconta. I don't believe that our textbook uses the phrase uniconta. They might. It doesn't show up in the lecture slides, but it does show up in that phylogeny that I gave you. In your phylogeny from the textbook, it doesn't show them as more closely related than any of the other groups. And again, this is because protists are a bit of a mess, and there's lots that we still don't know about protists and their evolutionary relationships. So amoebozoa are going to move by true pseudopodia. We saw the Phylos pseudopodia on the previous slide in the rhizarians. A true pseudopod or pseudopodia is going to be a cytoplasmic extension that pushes out from the cytoplasm. And then basically when this pushes out, or whichever direction it happens to be going, when it pushes out, the rest of the cytoplasm follows. And so it's basically kind of an oozing type movement. If you Google slime molds, they kind of look like almost like slugs that move forward. It's really kind of weird. They are kind of the animal-like, kind of the fungi-like. I would generally say that most of the amoebozoa are kind of fungi-like organisms. We have an example down here on the bottom of an amoeba forming a fungi-like structure, and then we have the slime molds here. You can see these little structures that are extending up. They're basically moving because of pseudopodia. So this one in the center is our slime mold example. Many of you might even think that slime molds are fungus because we use the word mold to describe a fungus. They share similar characteristics to fungi, but they are a different group because fungi into the opisthokonts group. Fungi and animals are more closely related to one another than either of them are to plants, but each one is separated into a separate group that distinguishes them. So we have fungi that are share a similar ancestor with animals, and we call this group the opisthokonts group. If we were to look at the amoebozoa, they come over here to a branch over here. Again, sometimes they're more closely related, categorized as more closely related than other groups, but it really just depends on where you're looking and the current research. So the opisthokonts are characterized by a posterior flagellum, one on a swimming cell. So that does not mean that all of our animal cells have flagella, but what it does mean is that an ancestor had a single flagella, and many of the organisms that evolved from that ancestor with that single flagella. And now we have these two groups, the fungi and the opisthokonts. The nuclearia are the opisthokonts genus that is most closely related to fungi. We're not going to say too much about them. We're actually going to move into fungi in the next lecture video, start talking about some of our fungal groups. But just know that they are the protist group that is most closely related. And really, I bring that up to demonstrate to you that our multicellularity evolved multiple times. So in the fungi group, we get multicellularity, but in the next most closely related group, the nuclearia, we just still have single-celled organisms. So we can see the nuclearia related to the fungi, and then over here, we've got a common ancestor with our animal kingdom, and this group called the Choanomonada. So the Choanomonada are the name of the group that is most closely related, and we are going to talk about a couple of specific organisms or organism types within that group. So those organisms within the Choanomonada group are called choanoflagellates. There are 125 species of opisthokonts protists. They all have this similar structure where we have basically a collar, and we'll see another picture of this on the next slide, a collar around one end and then flagella coming out the other. So these guys are kind of directional, and they are interesting because this collar allows bacteria to enter by water currents that are sort of swished through into a cavity on the inside of the choanoflagellates. So this is going to be important when we get to our earliest animals, and we'll talk about those guys in just a second. But our earliest animals are basically multicellular groups of these choanoflagellates. And the importance here with the collar is that each of these can bring in and filter out bacteria from the environment around it. And that's going to be important when these come together and we get our very first animal. So we've got a couple of examples of choanoflagellates here. We've got a single one here with that flagella coming out of the back. Water can come in, the collar is here on the side. Water can come in, filter, and it can bring in bacteria by phagocytosis. Here we have a different type of choanoflagellate, and then we have a choanoflagellate colony. So our earliest animals were actually probably some version of choanoflagellate colonies, and basically what they did is come together, associate with one another, and then some organism from their group ended up evolving into a sponge. And when we get to animals, we'll see that lining the inside of a sponge are all of these little organisms called choanocytes. Sorry, I misspoke. They are not individual organisms anymore. They are single cells, part of a multicellular organism. But you can see they're very, very similar. Cartoon drawing is similar to those choanoflagellates that we see here. And they come together, line the inside of the sponge. And so what happens is that all of these little choanocytes bring in food, basically filter feed food for the larger organism, which is this sponge. So we begin to see multicellularity back at the end of the Proterozoic eon. Our first animals are going to be invertebrate organisms. So when we get to chapter 26 and 27, we're going to talk about the evolutionary origins of our invertebrates. We're going to start with those choanocytes within our sponges, with sponges being the most primitive type of animal. They are multicellular organisms. Those choanocytes are now part of one of the cell types in those sponges, and so we'll come back to those. So our first animals are going to come out of this choanoflagellate lineage. They are generally going to have basically a direction. So they're going to be able to move in a particular direction. Many of them are going to have bilateral symmetry. You have bilateral symmetry. This is where you are the same if you draw a line down the center right to left, but not the same top to bottom. And this is going to, with our original choanocytes, having this type of directionality. They don't actually have bilateral symmetry, but they do have a symmetry to them. They do have distinct ends. And so this is going to be the precursor to our organisms that do have distinct ends, usually a head end and an anterior end. Our textbook has a really great summary table, table 23.2. This may or may not be in your study guide packet, but you can always look at it in the textbook. It's just got basically a list of the distinguishing features for each of these groups, and then some examples that we went over. We have smaller groups within the kingdom or the group that we have here. It does list them. I do strongly suggest that you make a lot of flashcards. This is a lot of memorization. You have to know a lot of specific things in each of our diversity chapters. So we've got chapter 23 with prokaryote and eukaryote diversity, and nothing is going to change when we get to plants and animals. Lots of studying. I suggest you start now, make lots of make lots of flashcards, and just really work on learning these names, learning these characteristics.